Welcome back, dear students. Today we will be talking about the structure of viruses. A complete virus or a complete virion, it is composed of two constituents, nucleic acid or genetic material, and this nucleic acid or genetic material it is surrounded by a protein coat which is known as capsid. Okay, here you can see this is the structure of a virus, or you can see here these are all viruses. Okay, the inner thing which is nucleic acid. Okay, and this nucleic acid is surrounded by a protein coat which is known as capsid. Now, first of all, we will discuss the nucleic acid type, the types of nucleic acids which are present in viruses. The nucleic acid in viruses, it can be either a DNA or an RNA. Usually, a virus which is of animal origin, it contains DNA as it is genetic material, while as plant viruses, they have RNA as their genetic material. Okay, or simply stated, you can say in majority of the plant viruses, the genetic material is RNA, while as in case of animal viruses, a majority of them contain DNA as their genetic material. And one more important thing is that the viruses, they contain either DNA or RNA as their genetic material, but they never contain both RNA as well as DNA. This is very much important. Now, depending upon the virus types, there are four categories of viruses based on the type of genetic material in them. Four categories of viruses based on the type of genetic material present in them. The first one is double-stranded DNA viruses. Those viruses which contain double-stranded DNA as their genetic material, and the examples are pox viruses like smallpox virus, cowpox virus, and chickenpox virus. Now, those viruses which contain single-stranded DNA as their genetic material, and the example is parvoviruses like gastrointestinal and lymphatic system viruses. The viruses which are found in the gastrointestinal tracts and lymphatic systems of animals, they contain single-stranded DNA as their genetic material. Now, double-stranded RNA viruses. Those viruses which contain double-stranded RNA as their genetic material, they are known as double-stranded RNA viruses. And the example is wound tumor virus in plants. These are those viruses which cause the formation of tumor-like structures in plants. Okay, and you have to remember that these viruses, they contain double-stranded RNA as their genetic material. All right. Then the fourth one is single-stranded RNA viruses. And the examples are tobacco mosaic virus, human influenza virus, and COVID-19. All these viruses, they contain single-stranded RNA as their genetic material. Now we are clear with the genetic material of viruses. The next thing that we have to deal with is the protein coat which is known as capsid. Here, this is the genetic material. This genetic material is covered by a protein coat, and this protein coat constitutes capsid. It protects the nucleic acid from degradation. Okay, it gives support to the nucleic acid. The capsid structure, the type of capsid structure, it is determined by the type of nucleic acid. What type of nucleic acid a virus contains, accordingly, it will have a unique type of capsid. Okay, different viruses have different capsid structures because they have different types of nucleic acids. Now the capsid, it forms the bulk of the virus. Since the genetic material of a virus is very much small in size, the bulk of viruses, it is constituted of the outermost capsid. And the the capsid, it is constituted of small protein subunits, and each subunit is known as capsomer. Here I can make it more clear. These are different types of viruses. Okay, say for example, we will here focus on the tobacco mosaic virus. It contains RNA as its genetic material, and this RNA, it is surrounded by these yellow colored subunits, and each yellow colored subunit, it is known as a capsomer. So a number of capsomers, they encircle the viral genetic material RNA, and the whole structure, this is known as capsid. Okay, so the capsid is basically a protein coat which protects the genetic material of the virus. Now the envelope. Now the envelope in viruses. It has been found that the capsid in some animal viruses, it is covered by an extra envelope, and that extra envelope is made up of a combination of lipids, proteins, and carbohydrates. Okay, the animal viruses, in some animal viruses, the capsid, in addition to capsid, there is one more outer covering, and that covering constitutes the envelope. And the envelope is constituted of, it is made up of a combination of lipids, proteins, and carbohydrates. And it has been found that some animal viruses, when they are released from the host cells, for example, this is a host cell, it contains a number of viruses. This is an animal host cell. When the viruses eject, or when the viruses come out of the cell, they are encapsulated by a portion of the host cell's plasma membrane, and this host cell's plasma membrane, this starts functioning as the viral envelope. Okay, this starts functioning as the viral envelope. Now the spikes are papillomers. Spikes are papillomers. These are basically carbohydrate and protein rich complexes which project, which emerge from the surface of the envelope. Okay, and these are found in case of HIV viruses, influenza viruses, and many others. And some viruses, they are known to get attached to the surface of host cells by means of these spikes. Here you can see this is the genetic material inside. Then we have this capsid protein coat. In addition to capsid, we have the envelope in case of animal viruses. And on the surface of this envelope, this is the genetic material, this is capsid. Then we have envelope, outermost covering in case of some animal viruses. And this outermost covering, it contains projections made up of carbohydrates and proteins. These projections are known as papillomers or spikes. The spikes are papillomers. They have been found to assist some viruses in getting attached to the surface of host cells. Okay. Now the enzymes which are present in case of viruses. So far, only a few viruses have been found to contain certain enzymes. The most important ones include, the first one is lysozyme. Okay, lysozyme, it is basically a muramidase or glycosidase hydrolase. Okay, it breaks the glycoside bonds. It is basically a muramidase or a glycoside hydrolase enzyme, and this enzyme has been found in case of bacteriophages. Bacteriophages are those viruses which infect the bacterial cells and lead to their lysis, which cause the death of bacterial cells. Okay, these enzymes break down the bacterial cell wall. For example, this is a bacteriophage inside a bacterial cell. It releases the enzyme lysozyme, and this enzyme lysozyme, it is effective against the cell wall. It breaks down the cell wall of the bacterial cell, and these viruses, they come out. Okay. The next type of enzyme that has been found in viruses is reverse transcriptase, and this reverse transcriptase virus has been found in some retroviruses like human influenza virus or human immunodeficiency virus. It has also been found in case of human T cell lymphotropic virus type 1, which is abbreviated as HTLV1. Okay, so reverse transcriptase enzymes, which help the viruses synthesize DNA from RNA, they have been found in HIV viruses, human T cell lymphotropic virus type 1. Okay, basically the function of a reverse transcriptase is it leads to the formation of a DNA strand using RNA as a template. Okay, so this was all about the structure of viruses, and under the structure we first of all studied the type of nucleic acids. Then we talked about capsid. Capsid is basically the protein coat that surrounds the genetic material or nucleic acid. And then we discussed that some animal viruses, they contain additional covering which is known as envelope, and that envelope is made up of proteins and carbohydrates. Okay, so with this we are done with the structure of viruses, and in the next part we have to deal with the prions and the related terms. Till then, stay blessed and thank you all.